Hello, my name is Nurhanizati binti Sharonza. My student ID is 2025888582 and I am from BE 148 Group 1D. Today's presentation is about plant propagation, which is sexual and asexual reproduction. So, what is plant propagation? Plants, there are two methods of reproduction, asexual and sexual. There are several methods of asexual reproduction, such as fermentation, budding, spore formation, and vegetative propagation. Sexual reproduction involves the fusion of male and female gametes, a process to continue the existence of plant species. Plant propagation is the process of multiplying or increasing the number of plants of the same species and at the same time perpetuating their desirable characteristics. What is sexual reproduction and asexual reproduction? So the definition of sexual reproduction is sexual reproduction involves the fusion of male and female gametes to form seeds. This process usually occurs through pollination and fertilization. Plants produced by sexual reproduction show genetic variation which improve adaptability to the environment, while the meaning of asexual reproduction is sexual reproduction produces new plants without the formation of seeds. The new plants are genetically identical to the parent plants. Common methods include cuttings, grafting, layering, and tissue culture. There are four types of plant propagation: asexual, which is vegetative. First, cutting. Second, layering. Third, budding and grafting. And lastly, tissue culture or micropropagation. So for the cutting, divide into three types: stem cutting, which is for softwood cutting, and the second one is leaf cutting. It's suitable for watery leaf type. And lastly, root cutting. The section of root are cut and placed vertically or horizontally and covered like to 3 or 5 cm. Then we got layerings, when the parts of plants propagated remain attached to their parents while rooting. There are two types of layering techniques. First, air layer, cutting, the most popular with roses and others. Second is simple layering or ground layering, flexible to branches of the old type of plants. Then we got budding and grafting. Grafting and budding are horticultural techniques used to join parts from two or more plants so that they appear to grow as a single plant. Types of budding and grafting. We got simple grafting, weed grafting, cleft grafting, approach grafting, bridge grafting, and bud grafting. So we move on tissue culture, which is micropropagation. It's a type of vegetative propagation. New plants grow very small pieces of plant material in aseptic condition, such as test tubes with a nutrient medium. Then quickly and can produce many new plants. It's virus-free and is now popular with orchid farm. What is actually tissue culture? Plant tissue culture is an asexual method of plant propagation in which small parts of a plant, tissues or cells, are grown in a sterile laboratory environment on a nutrition medium to produce new plants. It allows the rapid production of genetically identical and disease-free plants under controlled conditions. So next, we got the advantages and disadvantages of asexual reproduction. So we're going on to the advantages of asexual reproduction first. Firstly, the of asexual reproduction is produce plants with uniform characteristics. Second, maintains desirable traits of the parent plant. Third, faster growth compared to seed propagation. And lastly, suitable for plants that do not produce viable seed. The last thing is disadvantages of asexual reproduction: lack of genetic variation. Second, requires skills tools or controlled conditions. Third, plants may be more vulnerable to disease. And lastly, some methods are costly and time-consuming. That's all from me. Thank you for listening.